Next on the agenda is the reason you're here. Our speaker is Professor Dan Burke, who has come all the way from London today, this afternoon, about 3 o'clock, to be with us tonight. And he is an emeritus professor of pharmaceutical metabolism. He has a first-class honors science degree in biochemistry from London University, as well as a PhD in drug metabolism from the University of Surrey. 200 published research articles bear his name. We're looking forward to hearing from Dan Burke, and he is the other member of the Burke Potter team. Please give him a good hand, Dan Burke.
Thank you, Bob. Thank you, Bob, and thank you, Bain, and thank you, the Health Action Network Society, for inviting me over. Now, there's some technology here I've got to master. I've got a roving mic, so I'm not tied to the podium, and I've got a roving mouse, so hopefully this will go. I have to say that I'm really grateful for the opportunity. This is my first time in Canada, and you've given me some English weather today, I notice. I will apologize. I'm very croaky. I got on the plane, Zoom Airlines. Some of you probably know Zoom Airlines. It's not bad. Anthony Daniels, who is head of Nature's Defense and is a bit of a techno nerd, he said to me, you're okay, Dan. Zoom fly the most modern Airbus A300 aeroplanes. Well, he was half right. It's actually not a modern Airbus A300 I flew on. It's a very old Boeing 767-300. But it was a fantastic opportunity to come here. I'm really impressed by can I call you Hans? Is it upsetting? I read your paper, and as I was in the queue at Gatwick London Airport this morning, I got talking, as you do, to a lady behind me. Now, she's an English lady who has moved with her family out to Vancouver. And she told me about illness in her family. And so I said, look, you need to get in touch with these HANTS people, because they will give you the support you need. And I think this is what's so impressive about HANTS from what I've read. It's not just the educational events like this, but it's obviously the mutual support that you all give each other. I'm just sorry that I've got to make it such a short stay. I'm booked to speak in something called the Channel Islands. Some little islands. They are the UK territory, but then almost on the French coast in the English Channel. And I'm booked next weekend, so it's here and gone. I think I'd like to just set the context for what I'm going to talk about with a couple of... yeah, there we go, I've got it. So I don't think very many of you are likely to take issue with me on this. Fruit and vegetables combat cancer. The question is how? What's the mechanism? I'm going to give you a new mechanism. For some of you it's not new, but it's a different mechanism. It's not the antioxidant mechanism, it's a different one. And the other statement, which unfortunately is true, is that conventional anti-cancer chemotherapy has very major side effects. And the question one would want to ask is, can this be improved upon? And our answer would be, yes, it can, and we think we have a way of improving on it, and hopefully you'll see where I'm coming from as the talk goes through. It's a good idea, I think, to start with a definition. Salvestrols are natural chemicals. They are in fruit, vegetables, and herbs. They're activated inside cancer cells, and they kill cancer cells without harming normal cells. So the definition of a salvestrol is a two-part definition: the origin, where you find it naturally, and the function, what it does. What the definition of a salvestrol is not, it is not a chemical structural definition. There are hundreds of different sorts of chemicals in plants that can act as salvestrols. The thing that makes them salvestrols is what I call a molecular motif, a three-dimensional arrangement of a few atoms in that molecule that can, as it were, act as the key to unlock the activation mechanism. For it to be Salvestrol, however, something else has to happen, that having unlocked the activation mechanism, a potent cellular toxin has to result. And hopefully all will become clear as I go through. What I hope to do in the next 45 minutes then is to show you what these Salvestrols are, where they come from, what they do, mechanism of action, and then finish with something about why it's quite difficult to get enough Salvestrols in the natural diet, although it shouldn't be. So Salvestrols are in the natural diet. We can eat them very, very safely. It's highly significant that we find Salvestrols being prevalent in plants and diets that are traditionally said to prevent cancer, and diets and plants that are called traditional medicinal plants. Clearly, those who came before us knew a great deal. We are confident now that Salvestrols can help explain how plants prevent cancer and heal cancer. I certainly don't say that Salvestrols are the only mechanism. I'm a very strong believer in the redundancy principle, meaning more than is strictly necessary at any one time. Wouldn't you think it would be very dangerous if nature provided only a single mechanism of protecting our health? If that single mechanism failed, we'd have nothing. So I believe Salvestrols are one of many mechanisms for protecting our health. It's just that we seem to have forgotten almost all of them, and we're in the process of rediscovering them. And we think that Salvestrols have the potential to provide a treatment of cancer with far fewer side effects than traditional chemotherapy. And I really would like, before I go further, just to say something in defense of the pharmaceutical industry in this. Yes, and I used to tell the medical students at Aberdeen, cancer is the biggest failure of, it was 20th century medicine then, and it was. And until recently, it has been. And the conventional treatments that are available will continue to be. But the new molecular biology, the Human Genome Project, and all that sort of stuff, has revolutionized everything. The key to it are targets, molecules that are present in cancer cells and virtually absent or completely absent in normal cells. Once you have such a target, or a tumor marker, you can devise treatments. And there's a very famous cancer scientist called Karol Sikora in London. He has achieved an enormous amount in his professional life. And he said, months ago, that in his opinion, in 20 years, cancer will be a disease like diabetes. We may not be able to prevent or cure it, but we will be able to control it, such that being diagnosed with a cancer will not be a death sentence. Unfortunately, that is still likely to be about 20 years away. So we will not feel about, this is my view, we will not feel by then let down by the pharmaceutical industry in the same way. But there's a long time to go, and why not make use of the natural remedies that nature has provided us with? And we'll see what we can do. We go far. We perhaps get a little above ourselves on this one, but we think that Salvestrols change the paradigm of cancer health to one where the human body can defend and heal itself from cancer. And my good friend and my colleague Jerry said, and I think he's posed this to you in his talk, don't ask why do we get cancer. It's much more productive to ask the question, why do we sometimes not get cancer? And that is a new way. It's an upside-down way. Well, there's some pretty strong assertions I've made, and I think I need now to substantiate them. And to do that, I have to ask you, please, to come with me into the science of Salvestrols. I want you to think of the body's cells. These are those ectoplasmic bags that contain all our molecules and go together to make all our tissues, our organs, our structure, our bones, our blood. And every molecule we have and everything that goes on inside us pretty much goes on in the cells. And the point is that cancer is a disease of the cells. It happens when normal cells, which are producing just the right amount of the right chemical, occupying just the right amount of space, when they turn renegade, they run out of control and take over everything. And because cancer is a disease of the cells, then an ideal anti-cancer treatment is one that is highly specific to cancer cells and doesn't do much in the way of harm to normal cells. And this is the problem with the conventional anti-cancer treatments. They're not specific. And as many of you will know, they are beset by serious side effects. What I want to do now is take you through really our stock in trade, a type of experiment we do day in, day out when we're searching for Salvestrols. And the reason I want to do this is that it will show you, I hope, why we get excited about these things. We use human cells, cancer cells and non-cancer cells, and we keep them alive in these culture dishes and in culture bottles, on a gel, in a broth, simulating the body, and we treat the cells with a range of different doses of Salvestrols. And after about 3 days in growing in the presence of Salvestrols, we look down a microscope and we measure how many of the cells are still alive, how many of the cells are being killed, and we do that for each different dose of Salvestrol. And we plot out the graphs in this typical way. On the horizontal axis is the dose going from low dose on the left, high dose on the right. That's a logarithmic dose. It's a standard scientific way of condensing a very wide dose range. If it was your standard linear, it would be from one side of Vancouver to the other. So it condenses it. The point about it is, now I've been told there's a laser pointer on here. Yes, look. Every one of those major decisions is a 10-fold increase in dose. So 1 to 10, 10 to 100. It's quite an unusual type of axis unless you're in this area of science. And in the vertical axis, then, we plot the percentages of cells that are being killed. And it's a sort of upside-down axis because it's easier to understand. So no cells are killed, it's at the top of the axis, and 100% of the cells are killed, it's at the bottom of the axis. If we don't add any Salvestrol, by definition, we get 0 cells killed. And if we add a chemical and it isn't toxic to the cells, we would expect something like that red curve. No matter how high a dose of Salvestrol, you had no effect on the cells. But if we added a chemical and it was killing the cells, we'd see something like that blue line. The more Salvestrol you added, the higher dose, the more cells you killed. And what I'd like to do now is take you into Salvestrol Q40, we call it the first of our second generation Salvestrols. It's in 2 of our products. And what happens then if we do this experiment using Salvestrol Q40, adding them to cancer cells in culture? And that is the curve we get. And if there's any pharmacologists amongst you, you'll recognize this. This is a typical S-shaped curve, very typical of any compound that has a biological effect. And what it shows is that up to a certain dose, Q40 is not toxic to the cells. Then it gets to a threshold dose, and above that threshold dose, it is toxic. It kills the cells, and in fact, the higher the dose, the more the cells you kill. That's a nice curve. What I want to look at in more detail is zoom in on that blue area, which is the sort of active area. And so there's the curve again, showing that as you add more Salvestrol, higher dose, more of the cells are killed. But what happens if we do this experiment now with the non-cancer cells? And we get that. As you increase the amount of Salvestrol present, even when you get into those doses that kill large amounts of the cancer cells, you don't kill or even harm the non-cancer cells. At this point, we're getting excited. Eventually, a threshold is reached, and it is toxic to the non-cancer cells. But this is This is just illustrative of, it was Paracelsus back in the 1400s, I think, in Switzerland, he's the father of modern toxicology, who said everything is toxic at a high enough dose. And this is a non-specific blunderbuss effect. Paracetamol, salt, water even, they're all toxic in a high enough dose. And the key thing here is that you can never actually ever reach those levels of Q40 in the body. And I think you can start to see now what it is about these graphs that gets us so excited. It's the fact that the blue cancer cell kill curve is coming down way to the left of the red non-cancer cell kill curve. This is telling us that Q40 is able to kill cancer cells much more easily than it can kill non-cancer cells. And that is what we call selectivity. And just how selective is given by the distance apart. So we want those curves to be as far apart horizontally. Horizontally, because when you're thinking horizontally, you're thinking dose. And we want the selectivity to be as high as possible because we want our Salvestrols to be able to kill cancer cells as easily as possible relative to non-cancer cells. We like to put a number on this thing. Scientists always do. And by convention, to put the number on, we measure the selectivity, the distance apart, at the point where 50% of the cells are killed. And all we do is we look for the dose that kills 50% of the cancer cells and the dose that kills 50% of the non-cancer cells, and we divide one by the other, and it gives us a number. And the selectivity is 10. Now, that's a very high selectivity. There are actually not many types of drugs that are designed to kill cells. Anti-cancer drugs, I'm now talking pharmaceuticals, anti-cancer drugs and antibiotics. And of those, only antibiotics have anything like a 10 times selectivity. They have much higher. That's because bacteria are a different life form. With a selectivity of 10, you can be confident of this happening, that you've got such a margin of safety that you can find that a dose of Q40 that will kill a half to 3/4 of the cancer cells at a dose which is not leaving the normal cells unharmed. So the dose there, the black line showing killing 50 to 75% of the cancer cells, that same dose range in the non-cancer cells is leaving them unharmed. And it's this type of information, these data, that enable us to say Q40 is highly selective against cancer cells. And the aim with the supplements that we provide is to provide the right amount of Salvestrol so that the dose of Salvestrol you get in your body is in that zone of selective action, where the Salvestrol is doing harm to the cancer cells relative to the non-cancer cells. And one of the, this is supposed to be an update talk for those of you who have been to Jerry's earlier ones, and one of the updates I'm pleased to be able to tell you is we're just about that far away now from developing a blood test that can be used to measure the Salvestrol in the blood of people who use Salvestrol. We've got some logistics to sort out. Obviously, you won't want to be mailing them off to England, so we'll somehow do it through Vancouver here. But the idea is that both those who use Salvestrols know how much they're getting in their bodies, but we can help optimize the regimen. How often should someone be taking Salvestrol in order to make sure that they've got an effective dose in their blood for as large a proportion of the 24-hour day as possible. It's based on an area of science called pharmacokinetics, which gets quite mathematical but is very useful. So we're very close to that, and either Jerry or I or Brian Schaefer will keep you posted on developments on that. So how do they work? Why and how are Salvestrols specific against cancer cells? And it's, as Bob alluded to, it goes back to the 1990s. somebody said, I believe, writing novels is about 10% inspiration and 90% perspiration. And scientific research is like that, except you also need a few percent of luck in science. And this was our luck. We chanced upon this. Finding this enzyme, CYP1B1, is to all intents and purposes exclusively in cancer cells. And as I'll show you, the fact that it's an enzyme and the fact it's exclusive to cancer cells is the basis behind the Salvestrols that Jerry then went on to discover. Some people panic at this point. There are thousands of enzymes in the body. What do enzymes do? They make changes in the body's chemicals. You come across them in your biological washing powder. They make changes in the chemistry of the dirt so it's more easily washed out. But in the context of Salvestrols, you can safely and happily ignore every single enzyme there is except this one, CYP1B1. And I want to bring you back to a typical cancer tumor, prostate cancer, where the key thing is that the tumor is a mixture of cancer cells and non-cancer cells, which a pathologist can recognize. You can't actually see cells when you look down an ordinary microscope. What you have to do is stain their structures in order to see them. Now, what we have to do in our research is to be able to visualize CYP1B1, the Salvestrol activating enzyme, cell by cell. And, aha, is this echinacea? Right, thank you. Me and Pavarotti. Thank you. It's good. Thank you very much. So we had to devise a method. Now, what we start off by doing is treating the biopsy so that when we do whatever it is we do next, all the molecules stay in their allotted place. It's a bit like hard-boiling an egg. And then we take a very, very thin section through the biopsy. And again, that's like taking a slice through a hard-boiled egg. And when you look down on that slice, you can see the yolk in its place and the white in its place. And that's what we see. To see the CYP1B1 molecules, we devised a specific stain which drops a dollop of black dye or of brown dye on every molecule of CYP1B1. It uses an antibody to take it. It's called an immunohistochemical stain. And then to visualize the the cells that don't have the CYP1B1, we use that standard bluey purple dye I talked to you about. And what we see is typically that. Now, that's an example of a section through an esophageal cancer. The cancer cells are all staining black. A pathologist, again, will recognize those as cancer cells from their abnormal shapes. They're all staining black. That tells us that all those cancer cells have got this enzyme, CYP1B1. The normal cells are all staining bluey purple, which tells us that they don't have CYP1B1. Now, this, it's a type of cancer of the wall of the cervix, and it's an early stage before the cancer has broken out, and survival is very high if it's tackled at this point. It goes by the name of cervical intraepithelial neoplasia, and mercifully shortened to CIN. And CIN develops through 3 stages. Now, in the early stage of CIN, what happens is that a thin layer at the base of the epithelium, these are all epithelial cells, gets taken over by cancer cells. And then later on, that layer spreads to occupy about half of the epithelial cells. And finally, in the final stage, it is spread so far that it occupies all of the epithelial area. These are now all cancer cells, and the next thing that happens is it breaks out. When we stain these called colposcopy sections for CYP1B1, we find that the brown staining, we're using the brown version of the method, the brown staining mirrors exactly the spread of the cancer cells. And it's results like this, then, that lead us to be able to say that CYP1B1 has been shown to be present in all the cancer types that have been looked at so far, not just by our group, not just by my previous group, but groups in the America, in Japan, in a number of places. And not just in cancers, but in various stages of cancer. We now know that CYP1B1 appears at the very earliest stage of development of cancer and is also present in secondaries, metastases. In other words, at the very latest stage of development of cancer. It's there all the way through. We don't know why, but it's there. It's clearly there for a reason. I call it the seed of a cancer cell's own destruction, that we have been provided with this seed of destruction. We just need to know how to use it. And that list is already, actually, since I drew it up not long ago, it's already missing two. It's missing pancreatic and it's missing mesothelioma. So it gets bigger and bigger. People other than ourselves now are starting to call CYP1B1 a universal tumor marker. In other words, you could use it for detecting cancer, and we are doing some work on that, and as a universal tumor target. And some of you may know of the Dana-Farber Cancer Research Hospital and Institute in Boston. It's world famous. They're working with a venture capital type of pharmaceutical company in America, across the board. I always say America. What is it to you? Is it the States or them down there? They're working on developing an anti-cancer vaccine that is targeted at CYP1B1. And that's had a successful phase 1 clinical trial. But we went down a different route from this. But I need at this point just again to just stop and summarize and clarify. Jerry coined this term, the rescue enzyme. I think it's a very nice one. And just to remind you, it is only in cancer cells, but being scientifically correct, we say it's highly overexpressed. But this is crucial. It's not a dietary compound. CYP1B1 is not a dietary compound. CYP1B1 is not a food supplement. You can't take CYP1B1. CYP1B1 is an intrinsic component, for whatever reason, of your cancer cells. The one bit of enzymology that is useful to know about CYP1, however, is it's not just any old enzyme. It's a cytochrome P450 enzyme. What does this mean? It means that CYP1B1 can change the chemical structure of drugs. And when you change the chemical structure of a drug, you change its biological activity. And that led us to realize that we should be able to design something called prodrugs, targeted through CYP1B1 to target the cancer cells very specifically. And now this is the right time to introduce, or for some of you to reintroduce, to Jerry Potter, the younger, more photogenic half of the team. They don't make television programs about me. I'm a pharmacologist. I can do and interpret all the stuff I've talked about so far and most of what's going to come next. What I can't do is this. I cannot design and synthesize drugs. And I am at this point talking drugs, not natural compounds. Jerry is a medicinal chemist. He's a medicinal chemist of great renown. He has quite a lot of important anti-cancer drugs already under his belt, so to speak, before he came to join us. And so Jerry came and joined, and in no time at all, he had designed these prodrugs. What is a prodrug? A prodrug is a chemical that is totally harmless in the body until it is specifically activated, normally through an enzyme. And of course, what Jerry set out to do was to design prodrugs that are specifically activated by this CYP1B1 enzyme. And because that would only happen in cancer cells, these would then kill the cancer cells without harming normal cells. And he's been very successful, and these now have been sold on to a US, again, one of these venture pharmaceutical companies, and they're about to go into clinical trials. Now, I said, remember that science is 90% perspiration, a few percent inspiration, and some luck. Well, we've had our luck. Luck was discovering the CYP1B1. Now we come to the inspirational moment, or what I call Jerry's eureka moment. He said to me one day, look, we now know, through these synthetic pharmaceuticals that he's been designing, we know what the key motif is to open the lock of activation. He said, I bet you £1 to a penny, as we talk like that in England, I bet you £1 to a penny that we'll find natural chemicals in fruits and vegetable herbs that will also have this key motif. And they'll therefore work like the prodrugs, they'll seek out the CYP1B1 in the cancer cells, and they'll have this very selective effect against cancer cells. And of course, he was right, and we set to with a good team of PhD students and postdoctoral scientists. And the very first one we discovered was resveratrol, and you will all know about resveratrol, natural molecule in grapes and red fruit with lots of good health-giving properties and some specific anti-cancer properties. But our mechanism has nothing to do with antioxidants. In our mechanism, we showed that CYP1B1 converts resveratrol to pisiatanol pisiatanol was already known to be an anti-cancer toxin. It persuades cancer cells to commit suicide, apoptosis. And it is apoptosis, it's not apoptosis. Apoptosis is the modern Greek. And I tell you why I know it's apoptosis, because apoptosis was discovered at the University of Aberdeen, albeit a few years before I got there. But I did know one of the pathologists involved, and he told me that they asked the professor of Greek to give it a name. And it is to do, and the reason it's apoptosis is the picture of the cells as they're dying. It's like shedding leaves in autumn. And he called it apoptosis. So it's apoptosis. But the trouble with resveratrol is its dose-response characteristics, they're not really suitable for resveratrol. That's not to denigrate its action as an antioxidant, but just as a Salvestrol. I get my words mixed up here. As a Salvestrol, it's got a problematical dose curve. So we left resveratrol behind. Now what I want to do, and those of you who read your magazine will recognize this, but this is the moving picture version. This is the mechanism, basically, in cartoon form of how Salvestrols work. Here is a cancer cell with its molecules of CYP1B1, the activating enzyme. The resveratrol enters the cancer cell by no special mechanism. It diffuses in. It encounters the CYP1B1, and the Salvestrol gets activated to a metabolite. And I've just shown it in with a red atom just to indicate there's been a bit of a chemical change. Enzymes change chemicals. The key thing, though, is that metabolite of Salvestrol is the anti-cancer cell toxin, which then goes on to destroy the cancer cell. Whereas if you have a normal cell, the Salvestrol enters just the same, but there is no CYP1B1 for it to encounter, so it just goes back out again, and no harm comes to the normal cell. And that is the basis of Salvestrols. Now, if we look at on a wider scale, on a tumor, in this case it's an ovarian cancer sample, and once again, and we use the black version of the method, the cancer cells are staining black, telling us that they all have CYP1B1, and the normal cells are staining up bluey purple, telling us that they don't have CYP1B1. And just to remind you, tumors contain both cancer cells and normal cells. So in cartoon form again, here is a tumor. Cancer cells containing CYP1B1, the activating enzyme. Normal cells not. We flood the tumor with Salvestrols, and they enter both sorts of cells, both the cancer cells and the normal cells. But it's only in the cancer cells that the enzyme is there. So it's only in the cancer cells the Salvestrols become activated, and they selectively destroy the cancer cells. And that is what we're aiming to achieve. So this is that curve again. And now I think you've got the answer yourselves to the question, how can Salvestrols kill cancer cells more easily than they kill normal cells? The answer is that the cancer cells contain the CYP1B1 activating enzyme and the normal cells don't. But we went on, it was actually quite some time, it was a couple of years after we first did this experiment, to do what a scientist would consider the ultimate elegant proof. And we're very proud of this one, and I'm fairly sure you haven't seen this. We asked the question, what would happen if we do that experiment, but we now block the CYP1B1 in the cancer cells by adding a specific inhibitor? So we do the same experiment, but we add this additional chemical that specifically blocks the CYP1B1. And watch, the blue cancer cell kill curve shifts to the right and becomes indistinguishable from the red non-cancer cell kill. What this tells us is that when cancer cells have their CYP1B1 blocked, they lose their vulnerability to Salvestrols. Ergo, it is the presence of CYP1B1 that gives them that vulnerability. QED, and we went off to the pub to celebrate at that point. And we've gone on since then to test and find a range of Salvestrols. Here are some of them. With their selectivities, you know, selectivity, this left-right shift of the blue curve from the red curve. So Q40 selectivity is 10. T31G, which is present in our gold products, has twice the selectivity. It has another very important attribute. It is what we say lipophilic. It's fat-soluble. Means it can get across the blood-brain barrier. It can get into work at the brain, at the spinal cord. Fruit Force, our very first product. Now, this is interesting because it's primarily got Q40 in it, but it's got 3 times the selectivity of just Q40. This is measured in those cell experiments. This is not in people. This is in the laboratory. And I think it's the fact there are other natural compounds from the variety of fruit. It's just the freeze-dried extract, which are synergizing the Q40. You'd expect that. People who understand natural remedies would not be surprised. And then P55, one of our third generation. That's not a misprint. That selectivity is 23,000. That is as good as the best of Jerry's synthetic pharmaceutical prodrugs. It's quite outstanding. These are the curves for Salvestrol P55. Look at that separation. And what it shows us that P55 can kill over 3/4 of cancer cells at doses that leave 4/5 of the normal cells unharmed. It's not no harm comes to the normal cells, the non-cancer cells. It's 4/5 of them. Sometimes are unharmed. Sometimes there is an effect. But the difference between the effects is enormous. And with having that kind of selectivity, the right-left shift there, you've got an enormous safety factor that if you were to accidentally take more than you meant to, you've got to take hundreds of times more than you meant to before you start causing real harm to the normal cells. And I'll tell you a little bit more about P450 towards the end. Oh, here we go. That's well, I've talked that through. Cool. So, do Salvestrols work in people? I don't know what the law is here in Canada. I should have checked with Brian. But in the UK, I can't make a claim, a clinical claim that Salvestrols work against cancer. The law forbids me. There's a 1933 Act of Parliament, pardon me, very important. It's designed to protect people against charlatans. But it says that there are only 4 categories of professional who are allowed in law to make a claim that a product has some use in cancer to a lay audience. I can talk and I'm not a medical doctor, I'm not a pharmacist, I'm not a nurse, and I'm not an MP. Members of Parliament have this right. I can make those claims to practitioners, and you have naturopaths and NDs here. I think it's assumed that someone who is a professional practitioner has enough knowledge to decide whether I'm talking baloney or not. But it protects the lay public. However, I do believe the law allows me to let Salvestrols speak for themselves. And what I want to say is we have a growing file of signed testimonials from users telling us that they actually really want to tell us that these things work. Everything from relief of pain to a few extra months, anything from 1 month to 6 months of life, beyond when they were told to die. And I find that the most poignant. That's the one that affects me most of all when I meet these people. A number of people have written to us and said that according to clinical tests, their tumours have shrunk and they have ascribed this to their use of Salvestrols. And other people have actually said it has cured the cancer completely. I have to stress, these are not medical people. They're only patients. And what do patients know? Someone pointed out to me, they could be friends and relatives who we've asked to write in. So it's not a clinical trial. We haven't got a clinical trial. But for my money, it is evidence growing that Salvestrols do have an effect in people. Please forgive me, I'm not putting these testimonials on the screen, nor am I going to read them out. We are working on some way whereby people can look at it. But the key thing, it's been pointed out to us, we must do is try and get the medical doctors of these people, even off the record, to affirm that what has been written in the testimonial is true. Let me move on and start summarizing. The activating enzyme, the CYP1B1, it's in all types of cancers. It is a generic cancer prevention and rescue mechanism, and it is a mechanism based on natural food. And I think of it as a Trojan horse, sat there quietly doing no harm in the cancer cell until you feed it Salvestrols. And then what it releases is not a Greek army, but a horde of Salvestrol-based anti-cancer toxins. And as I said before, either through evolution or by divine design or whatever you want, it would appear that we have been provided such that in CYP1B1, the cancer cell has a seed of its own destruction as long as we know how to make that seed grow. Now I want to introduce something that is an update. And this is quite hard, so forgive me if I lose you. Because of the internet, users and patients become very, very knowledgeable about their diseases and the cures. And of course, well, certainly in the UK, the doctors hate this. And quite a lot of you may well have picked up by now that CYP1B1 in different people can undergo a genetic variability. And that some of this variability seems to result in a disease of the eye called primary congenital glaucoma. And the question that has been put to me is, will people with glaucoma in the West, in Canada, in Europe, in America, will people who have glaucoma therefore have an aberrant CYP1B1 and not benefit from Salvestrols? I'm going to show you that the answer to that is no. You probably remember DNA makes protein. In this case, a gene for CYP1B1 makes CYP1B1. That's a molecule of CYP1B1. It's actually a close cousin of CYP1B1. I couldn't find a picture of a molecule of CYP1B1. And in the middle of it is a yellow heme moiety, which is the business end that does the activating. Now, the adage, the thing that Nobel Prizes were won for, is that DNA codes for, if you like, makes protein. And what I've shown you here is is a little bit of DNA, and it's to show you, to remind you, because you probably all know, that DNA, the genetic code is a series of words, and they're 3-letter words from a 4-letter alphabet. So the 4-letter alphabet are called A, T, C, and G. And if I go back one, the A, T, C, and G are, if you like, the steps of the spiral staircase. They're nucleotides. So it's a 4-letter alphabet which makes 3-letter words. And I've got 3, for example, words here. We call the words codons, but it's as easy to think of them as words. So if we look at that the first word there, the first codon is TGC, and that go the right way. That makes an amino acid. It causes an amino acid to arise, which then becomes part of a protein. If we look at the next word, the next codon, that's AGC, and that also now produces an amino acid. It's a different amino acid, and amino acids are the building blocks of our proteins. They go together to make proteins. And the bottom codon, the bottom word is TAC, and that goes on to generate yet a different amino acid, and there's a little tripeptide. Okay. Now, this is the bit that gets really hard. What this is all about is something called genetic polymorphism. And I think the easiest way is to sort of follow me through it rather than darting about. Genetic polymorphism, also known as SNPs or SNPs, single nucleotide polymorphisms. And if you read Science magazine and the like, you will have come across SNPs a lot. And all that means is a single nucleotide polymorphism. I will show you. This is what I had on the previous slide. There's the same 3 words and the same 3 amino acids. Now, what happens when you get a SNP is that one of those letters in the word changes. So here, word 1, codon 1, is no longer TGC, it's now become TAC. And that is a single, because it's happened to one letter, nucleotide, because these are nucleotides, that's what they are chemically, polymorphism, because it's caused 2 different versions of the word to occur. Polymorphism means in different forms. If there are any pharmaceutical scientists amongst you, you will use polymorphism to mean different shapes of crystals rather than different types of genes. So this is what's going on, and it's a SNP. And so this is happening in all of us. If we look at our genes for CYP1B1, we find a number of these exchanges. The result of that change, of that single letter, G being changed to A, is that it results in a very different amino acid. You see, the amino acid up there from TAC is a different amino acid from TGC. This, then, if you extend this out to a full protein, is a very different protein from that. That is enough of a change to alter the characteristics of the protein. And if that protein is an enzyme, if it's CYP1B1, that change of one letter of one word in that whole genetic code is enough to change the ability of CYP1B1 to work. Now then, this is really coming to this, to try and help you understand what I'm saying here. Because this is the good news and the bad news bit. What then is the story with polymorphism in CYP1B1? Polymorphism, again, merely meaning your protein, your enzyme, can exist in more than one variant form. There are 50 or more rare mutations of the CYP1B1 gene that have been recognized. They all seem to result in primary congenital glaucoma. This is a very rare disease. It's most common in Southeast Asia and the Middle East. It is very uncommon in Europe, in America, in Canada, etc. And these mutations, which occur very rarely, obliterate the enzyme activity of CYP1B1. So that if you suffer from one of these mutations of the CYP1B1 gene, it is unlikely, most unlikely, that you can activate a Salvestrol. It is unlikely that you can benefit from Salvestrol, but it is highly unlikely that you will actually have one of these mutations. It is entirely different from the type of glaucoma that we see in what we call the West. The type of glaucoma we see in the West has an onset of about 50 years of age. Primary congenital glaucoma onsets in the very young, 2 years, 3 years of age. The type of glaucoma that we see, that some of you may well have, does not involve the CYP1B1 gene. So if you are unfortunate enough to have the type of glaucoma we see here, it does not affect your ability to benefit from Salvestrol. However, there are 4 common polymorphisms of the gene, not 50 plus rare ones, but 4 common ones, which can affect up to half a population. And this is interesting. These actually don't significantly change the activity of CYP1B1. You get a very small increase or a decrease. And all the scientists who've looked at this have all said this is not a meaningful increase or decrease. What is coming out, though, from these is that these are associated with a significantly altered risk to certain types of cancer. And this story will develop over the next few years in a very interesting way. But I do this so that you don't need to worry that if you have the Western form of glaucoma, the evidence is that there's nothing to do with the CYP1B1 gene. And this I show you so that you can keep an eye on it. For example, there are 2 particular ones of those SNPs involving 2 particular amino acids at certain positions in the CYP1B molecule, 119 and 432, alanine to serine, valine to leucine, that seem to be associated with a markedly different risk to breast cancer, to prostate cancer, and interestingly, to the risk of prostate cancer becoming aggressive. But there's a long way to go on these. Summarizing then the Salvestrols, natural plant compounds, CYP1B1 rescue enzyme, metabolize the Salvestrols, happens only in the cancer cells, so the cancer cells are killed and not the normal cells. So now, can I convince you perhaps that the answer to the question I posed earlier, why do we sometimes not get cancer? Maybe the answer is, well, because sometimes our diet contains Salvestrols. But all in the garden is not rosy, because it turns out it's very hard to get enough Salvestrols from a modern natural diet alone. All sorts of plants are high in Salvestrols. Lots of herbs, chaparral, dandelion, burdock, thyme, rosemary, they're high, or they should be high. Aubergines, peppers, red, green, orange peppers, lots of citrus fruit, cranberries, strawberries. But when we started measuring the levels of Salvestrols in a lot of these foods, we found that there virtually weren't any. And the reason is that modern agricultural practices have depleted our food of Salvestrols. And there are 3 main reasons. Salvestrols are produced by plants, not, we think, because of anything to do with cancer, but because Salvestrols are part of the plant's own defense mechanism against pathogens. When a fungus infects a plant, it sends a chemical signal to the plant, eliciting, as the botanists say, eliciting the synthesis of Salvestrols. And it actually gets even more interesting than that, because different fungi on the same plant will elicit different Salvestrols. Farmers don't like their crops to be infested with fungi, so they routinely spray them with synthetic agricultural fungicides. So now the plant never gets infected with a fungus, and it never receives the signal to synthesize Salvestrols. And so we find that Salvestrols are truly much higher in organic produce. The second reason is you and I. We like our fruit juices to be sweet. And we like our olive oil to be clear. And what we find is that the manufacturers are deliberately removing the bits and the bitterness. So cranberry juice should be a very good source of Salvestrols. And we went into the manufacturing plant of a major cranberry juice maker in the UK. And we walked in, and what we saw was like a huge chemistry lab. Why? Why would a cranberry juice factory have a chemistry lab? The chemistry lab were the ion exchange columns used to extract the bitterness out of the cranberry juice. The government, certainly the UK government, quite understandably, doesn't want manufacturers putting sugar in to disguise the bitterness. We don't want the bitterness, so they take out the bitterness without realizing that that's where the Salvestrols are. Salvestrols are bitter. So that brand of cranberry juice had virtually no Salvestrol. When we tried and tested a good, natural, whole food, healthy, organic cranberry juice, then it was bursting with Salvestrols. Olives are an excellent source of Salvestrols. But in olive oil, you'll only find them in the cold-pressed, cloudy, unfiltered olive oil. That's all we use at home. Not in that very sparkly stuff. Why? Well, because the Salvestrols are in the bits. So you'll only find Salvestrols in unprocessed whole foods, not in the modern processed foods. And the final reason why our diets are depleted, again, is this sweetness. We like sweet fruit. We like our fruit to be sweet. Salvestrols contribute to the bitterness. And so over years, the fruit breeders have bred fruit varieties that taste sweet. And in doing so, they have lowered their Salvestrol content. And we at Nature's Defense are doing research with growers of traditional fruits, especially the apple growers, and with two of the UK's major publicly known research organisations, Kew Gardens, which is a very prestigious horticultural and botanical research organisation, and something called the Eden Project. And if any of you are ever in the county of Cornwall, which is in the sort of long, thin bit down to the southwest, go to the Eden Project. It's a smashing thing. They've built an artificial sort of tropical plant paradise there. But they see as part of their mission the education of the population. And we're researching with the Eden Project. So what we're trying to do is see if we can go back to older varieties of fruit that might have higher levels of Salvestrol, which then... this used to be the last slide, but there's one more especially for this talk. Are there benefits then of a Salvestrol diet supplement, a food supplement, when our modern diet is seriously depleted? We obviously think there are. I would put it to you that the supplement benefits from our specialist knowledge of which are the best Salvestrols and where can we find them, and they guarantee an intake of the necessary Salvestrols in a very convenient form. And just to bring you, on the last slide, slap bang up to date, we're now designing a brand new Salvestrol formulation. It will have all three of the Salvestrols I've shown you, Q40, T31G, and a new 1P55. We'll also have in it biotin, which has been shown to elevate the levels of CYP1B1 in cells. So biotin should accelerate the activation process. Niacin and magnesium will be in it because these stimulate the activation process, the enzymatic reaction. When will this be available? All I can say is it's in development, but Brian Schaefer will have it when it becomes available. And I really just want to thank all of you for bringing me over and giving me the opportunity, even on a dreich day, as we used to say in Scotland, to appreciate Vancouver. And thank you very much.